Quantum life: how is biological information encoded and how is it inherited? We always take the ability of organisms to accurately copy their own genomes as a matter of course, but this ability is the most extraordinary and fundamental characteristic of life. The error rate of DNA replication, which we call variation, is usually less than 1/109th. In order to give us some concepts about this shocking accuracy, we can imagine the hundreds of thousands of words, punctuation marks and spaces included in this book. Now, suppose there are about 1,000 similar books in the library. Book, your task is to faithfully copy every word, punctuation and space in these books. How many mistakes do you think you will make? This is exactly what the medieval scribes did before the invention of the printing press. They must do their best to copy the text by hand. The texts they copied were full of mistakes, which is not surprising, as we can see in the different manuscripts full of differences in the Middle Ages. Of course, computers can replicate information with very high accuracy, but computers rely on modern electronic digital technology based on solid hardware to complete tasks. Suppose you make a copier with wet and sticky material. How many mistakes do you think if you use this machine to read and write the content to be copied? However, if the wet and sticky material is a cell in your body and the information to be copied is encoded in the form of DNA, the number of errors will be less than one word, which is 1/109th. High-precision replication is essential to life, because the high complexity of living tissue requires that the setting of instructions must be equally complex, and every error can be fatal. The genome in our cell is composed of about 3 billion gene letters and encodes about 15,000 genes. But even the simplest self-replicating microorganism has thousands of genes in its genome, written by millions of genetic letters. Although most organisms can tolerate a very small number of mutations in each generation, allowing the next generation to have more than one number of mutations will cause serious problems, such as the formation of human genetic diseases, and even the birth of unviable offspring. In addition, whenever cells in our body replicate, including blood cells, skin cells, and other cells, we must also replicate the DNA in the cells and inject them into the daughter cells. If something goes wrong in this process, it can cause cancer. Why quantum mechanics is extremely important to genetics. To understand why quantum mechanics is so important to genetics, we must first return to Cambridge University in 1953. On February 28, 1953, Francis Crick stormed into the Eagle Bar in Cambridge and excitedly announced that he and James Watson had discovered the secret of life. Later that year, they jointly published a historic paper, which showed a structure and gave a set of simple rules to provide answers to the two most fundamental mysteries of life: how biological information is encoded, how is it inherited. In many documents, when describing the discovery of the genetic code, it is customary to emphasize an arguably less important feature: DNA has a double helix structure. This discovery is indeed striking. The beautiful structure of DNA deserves its name, and thus it has become the most iconic image in the scientific community, appearing in T-shirts, web pages, and even architectural design. But the double helix structure is essentially just a scaffolding. The real secret of DNA lies in the material installed on the scaffolding. We have briefly introduced that the double helix structure of DNA is supported by a sugar phosphate backbone, which carries the true information of DNA, nucleic acid base chains, including guanine, G, cytosine, C, thymine, T, and adenine, A. Watson and Crick discovered that the linear sequence of bases constitutes a certain code, and they believe that this is the genetic code. At the end of their historically significant paper, Watson and Crick stated that the structure of DNA also provided a solution to the second biggest mystery of life. They wrote, "This has not escaped our attention. The proposed pairing hypothesis also provides a possible mechanism for the replication of genetic material." What has not escaped their attention is an important property of the double helix structure: the information on one of the double strands of the helix, or the base sequence, can be regarded as a complementary copy corresponding to the base sequence on the other chain. A on one chain is paired with T on the other chain. Similarly, G is always paired with C. The special pairing of bases on the corresponding chain, AT pair or GC pair, is actually caused by a weak chemical bond, called hydrogen bond. Like a glue, the hydrogen bond that binds two base molecules together is essentially a hydrogen atom shared between the two molecules. Hydrogen bonding is important to our story, and we will discuss its nature in more detail later. The weak binding between the double strands of paired DNA naturally provides a mechanism for gene replication. The double strands are untwisted into two single strands, and each single strand can be used as a template to form a complementary new strand on its own basis. Make the original double strand into two, and complete the replication. This is exactly the process of gene replication during cell division. After the two strands of the double helix structure and the complementary information they carry are unwound and separated, an enzyme called DNA polymerase is brought close to the separated single strand. Subsequently, the DNA polymerase combines with a single strand and slides along the nucleotide chain of the strand, reads each genetic letter, and inserts the complementary base at the corresponding position with accurate accuracy, so that new strands are gradually formed. DNA polymerase inserts AT at the corresponding position whenever it encounters A, and inserts AC whenever it encounters G, until a complementary single strand is completely formed. The same process also took place on another single strand just separated, turning the original double helix into two, one in each daughter cell. This seemingly simple process constitutes the basis for the proliferation of all living things on our planet. However, Schrödinger insisted in 1944 that the surprisingly high accuracy of genetic processes cannot be explained by classical laws of physics. He believed that genes are too small and the regularity of genes cannot be based on the principle of order from disorder. Schrödinger proposed that genes must belong to some kind of aperiodic crystal. So, are genes really non-periodic crystals? Are genes really non-periodic crystals? Crystals, such as salt granules, have their special morphology. Sodium chloride crystals, ordinary salts, are cubes, and water molecules in the form of ice form hexagonal cubes and form snowflakes in various forms. The shape of the crystal is the result of the orderly accumulation of molecules inside the crystal. Therefore, in the final analysis, it is the quantum law that determines the shape of the crystal, because the quantum law determines the shape of the molecule. However, although standard crystals are highly ordered, they cannot encode information. Because each repeated crystal unit is exactly the same, a bit like a checkerboard pattern wallpaper, a simple rule is enough to describe the entire crystal. Schrödinger proposed that genes belong to what he calls aperiodic crystals, that is to say, such crystals have a repeating molecular structure similar to standard crystals, but after some adjustment, there are different intervals or periods between repeating units, so-called aperiodic, or the repeating unit itself has a different structure, more like a tapestry with a complicated pattern than a wallpaper. Schrödinger believes that these adjusted repetitive structures encode genetic information, and like crystals, their order should also be at the quantum level. Note that Schrödinger made these claims 10 years before the discovery of Watson and Crick. The structure of genes, and even the composition of genes, became known gradually after many years. So, is Schrödinger right? Obviously, the DNA code is indeed composed of repetitive structures, DNA bases. DNA bases appear acyclically, and each repeat unit must contain one of four different bases. As Schrödinger predicted, genes are indeed acyclic crystals. However, non-periodic crystals do not necessarily encode information at the quantum level. For example, the irregular particles on the negative of the photo are caused by silver salts, not quantum phenomena. In order to test whether Schrödinger's prediction that genes are quantum subjects is equally correct, we need to look more closely at the structure of DNA bases, especially the complementary base pairs between A and T, and C and G. DNA pairings that carry the genetic code rely on chemical bonds that bind complementary bases together. As we have mentioned before, these bonds are called hydrogen bonds, which are formed by two atoms sharing a proton, that is, a hydrogen nucleus, and the two atoms belong to the complementary bases on the corresponding two single chains. Exactly these hydrogen bonds allow base pairing. Base A is paired with base T, because each proton on A is exactly in the right position and can form a hydrogen bond with T. Base A cannot be paired with base C because the protons are not in the correct position to form hydrogen bonds. The nucleotide bases paired with protons are the genetic code that is copied and transmitted between generations of life. Moreover, this is not a one-time transfer of information. It is not information encrypted with a one-time password book, and it is destroyed after use. The readability of the genetic code must be able to run through the life of the cell, so as to direct the cell to complete the protein production process, create the engine of life enzymes, and use enzymes to organize all other cell activities. This process is done by an enzyme called RNA polymerase. Like DNA polymerase, RNA polymerase reads the position of protons encoded along the DNA strand. Just as the meaning of a message or the layout of a book is determined by the position of words on the page, the position of protons in the double helix structure determines the story of life. For more information on your true calling is encoded in your DNA. Please go to the below link now and don't forget to push the subscribe button below.